Hey guys, this is Miss Baker. Today I wanted to talk to you about a really important process that's going to kind of bridge the gap for us between the biosphere and the hydrosphere. So we've spent this first 3 or 4 weeks talking about the biosphere, bio meaning life, so the living part of the earth. Next week we're going to transition into the hydrosphere. So this doesn't particularly have to do with what we're talking about with human population this week, but it's going to kind of bridge the gap for us. And that process is the nitrogen cycle. So nitrogen is not the most abundant element in living things, but it is an important element in living things. It is a part of proteins. So really important. So how do we get it? Well, first of all, the atmosphere is made of about 78% nitrogen. But somehow it has to get into a usable form for us. And that process is called nitrification or denitrification. So nitrogen in the atmosphere can be converted into ammonia through several different processes. Lightning striking can do it, or there are bacteria that live in the soil that can actually do this process. They make it where plants can use it. Plants then produce proteins using the nitrogen. Animals like us eat the plants and then use the nitrogen. Then animals produce waste products, and those wastes are high in nitrogen. So then that nitrogen can also can be added back into the system, and it can be changed by more bacteria and either released back into the atmosphere or put back into circulation with plants. So I'm going to stop here and add in a picture of the nitrogen cycle for you. So I am working in the school greenhouse today to kind of show you the nitrogen cycle in action. So you know that we have crawfish and we have plants growing here in our aqua center. Like I already said, animals produce waste. That waste is high in nitrogen. It's high in a type of nitrogen called ammonia. Ammonia is not usable by plants. So there's bacteria living in the water that takes the ammonia and turns it into nitrites. Nitrites then can be used by the plants to produce proteins. So we are going to look at some water samples and see what the ammonia levels and the nitrogen levels look like in our tanks. This is the first tank that we're going to test. You can see that it has plants growing in it, and it also has cages with crawfish in it. Now, during the hurricane just 2 weeks ago, we had quite a few of these crawfish actually die. So we should have had then a high amount of nitrogen or ammonia in the water. However, these plants are growing really, really well, so that tells me that there were also bacteria that converted the ammonia into nitrites, which the plants could use. So now I'm going to take a couple of samples of water and come back and test them. Alright, so I have my 2 water samples. I have 5 milliliters in this test tube and 2.5 milliliters in this test tube. I'm going to use the 5 milliliter test tube to test for ammonia. Remember, ammonia is something we do not want. And I have instructions here for what to do. First, I need to add 10 drops of salicylate ammonia reagent number 1. Remember, lab safety, you should always have goggles on. Squeeze the tube, turn upside down, let go so it will make a vacuum. and put it where you can see it. Add 10 drops. And I'm going to cap that and mix it. The next step is to add 7 drops of reagent number 2. I'm going to form a vacuum just like I did with reagent number 1. And the next step is to add 7 drops of reagent number 3. See, it's already starting to turn kind of a yellowish color. And this needs to wait for 20 minutes. So I'm going to set a timer for 20 minutes. Set a timer for 20 minutes.
Okay, 20 minutes and counting.
Alright, so that's my ammonia test. Now I'm going to test for nitrite. Ammonia bad. Nitrites are good for plants, but if we already have a lot of plants, probably our nitrite is going to be low. So we'll see. I am going to dilute this with mixed acid reagent. And it's currently at 2.5 milliliters. I'm going to bring it up to 5. Cap that, mix it, and then I'm going to add a 0.1 gram spoonful of color developing reagent, which is this right here. Here's my little spoon. I'm going to cap and mix this. You want to just keep tilting it till it's totally dissolved. Got kind of a chunk going in there. Alright, and this one, we have to wait for 5 minutes. So I'm going to set it aside for 5 minutes. I am going to go ahead and repeat those 2 tests for 2 other tanks. And I'm going to take you to show you. Great, tank number 2 is a much larger tank. It has the most amount of crawfish in it, and it has also a little bit more plants than is in tank 1, but I would say for the size, it has less plants in it. Tank 3, kind of in between the sizes of tanks 1 and 2. There are crawfish living in here, but there are less crawfish than are in the other tanks. There are a few plants, but they have not been growing as well as the plants in the other tanks. So I'm curious to see what the nitrogen and ammonia levels are like in this tank. Right, I am going to go ahead and test tanks 2 and 3 for ammonia at the same time. I'm trying to do these as close together as I can so we can compare them to each other. Start with 10 drops of number 1. Seven drops of number 2. And 7 drops of number 3. And again, I'm going to set these over here to wait about 20 minutes. And so I'm going to grab another timer and set for 20 minutes. This is tank 1 that's been sitting for a few minutes already. Tanks 2 and 3, they are all actually changing pretty quickly, which tells me that my ammonia levels may be too high. I don't know if you can see, but tank 3 is already really turning. That's really surprising. All right. Put those back. And I'm going to grab these 2, which I'm going to, this is tank 2 and tank 3. And I'm going to test these for nitrites. Nitrites good, ammonia's bad. I'm going to fill these to the 5 milliliter line. with mixed acid reagent. I know you guys cannot see my other test tubes, but I am amazed in what a short time those two have already started to show some major problems with the water quality. And I really thought that those two tanks were fine, and the other one was having a problem. So that's interesting. The water, we've had lots of algae in tanks 2 and 3. I'm not sure what's exactly going on in there. Right, my mixed acid reagent is now up to 5 milliliters. Going to shake these up. And I'm going to add 1 scoop of color developing reagent to each of these. Mix these up. And already see there's starting to be quite a difference in the two tubes. Set these back. And for my first tank, tank 1, my nitrite level, that test that takes about 5 minutes, it's been longer than 5 minutes. And you can see the color here. Not sure how well you can see it. Let me just grab a piece of white. So a very, very faint light pink would be not much nitrite. A dark pink would be a lot of nitrite. This is a good amount. Seems reasonable considering that we do have things growing in there, and the plants are doing very well. So we grab back these tubes for ammonia. Tank 1. Tank 2. And tank 3. Tank 1 here, very light in color, that's very good. Tanks 2 and 3, very dark in color. We've still got 14 minutes out of 20 minutes left on these last two. That tells me there are big problems with those two tanks. We do not have the nitrifying bacteria that we need to change this ammonia into nitrites. So, what can we do about that? Well, the only thing you can do is grow more bacteria. how do you grow more bacteria? Well, if you put more surfaces in for the bacteria to grow on, they need lots of surface area. Another problem could be the algae maybe outcompeting the bacteria for resources or for space, so we can try to eliminate the algae as much as possible. We've already been trying to do that with those floats that are on top of the tank to block sunlight. Also, it's getting to be colder out, so it's autumn now, so we'll have less sunshine, so that should help with our algae. But also, we believe there's a way to just buy the bacteria, so we're going to be looking into that soon. All right, we've got a few more minutes on the nitrites, so I'm going to come back to those in just a couple minutes. Let me see. Actually, we're probably good to go ahead and check those, because I was at about 6 minutes when I said that, and now I'm at about 10 minutes, so pretty close. I'm going to put 1, 2, and 3 back over here. I'm going to go ahead and grab my nitrites, which are good for 1, 2, and 3. So if we have high ammonia, that's going to mean we don't have high nitrites. We don't have enough bacteria changing the ammonia out of ammonia and into nitrites, and that's exactly what we see here. Tank 1, which was good on ammonia, is good on nitrites. Tanks 2 and 3, which were not good on ammonia, their ammonia was too high, their nitrates are too low. And now this is a little bit more subtle color change than what you see in the ammonia, but you can definitely still see tank 3 is the lightest and tank 1 is the darkest. So that's super interesting, sort of, but what does it have to do with the biosphere and the hydrosphere? So what does nitrogen have to do with the relationship between the biosphere and the hydrosphere? I'm glad you asked. I'll tell you. There's a process called eutrophication that we talked about when you were in the 9th grade, but you probably don't remember. Eutrophication is a process where too much nitrogen gets into a body of water, usually the ocean. The way that it enters the ocean is through the watershed, so it washes from the land into the ocean. So where could this nitrogen come from? It could be from animal waste, like what we just saw in our tanks here, but more often it's actually from fertilizer. So if you think about the United States, think about the Mississippi River. Kind of goes right down the eastern middle part of the United States, and it is surrounded by farmland. All of that farmland is either used for cattle, pigs, chickens, turkeys, things that produce waste, or it's plants that are fertilized. All of that fertilizer, when you put too much fertilizer, it then washes into streams and rivers and eventually makes its way into the ocean. Well, fertilizer, that sounds like a good thing in the ocean, right? It's going to do what fertilizer does. It's going to make plants grow. The major plant, not really a plant, but the thing that uses nitrogen in the ocean is going to be algae. And we get something called an algae bloom. I'll insert some pictures here of algae blooms. Okay, so we've had an algae bloom. We have lots of algae. Still doesn't sound that bad. It's food for things, right? Well, it's okay for a little while, except not really, because some algae actually produce toxins in the water. But even if they don't produce toxins, whenever they bloom like that, eventually they die. So we've been talking about population growth graphs this week. As population grows exponentially, it can't keep that up forever. Eventually, it's going to crash. It's going to boom and bust. When it crashes, then it is eaten by decomposers, like bacteria. Not the good kind of bacteria that we have been talking about, but bacteria that, like all living things, use oxygen. So the bacteria start multiplying, and they are sucking all of the oxygen out of the water. So what does that do to other organisms that need oxygen? Oh, Siri's trying to tell me about it. She thinks I'm asking her, not asking you. But it sucks that oxygen out, and things like fish that use oxygen die, and you'll have dead fish float to the surface when you have algae blooms. So those are major problems. There's a spot just off the coast of Louisiana called the Gulf Dead Zone that basically nothing can really live there because the nutrient levels are too high, and therefore the oxygen levels are too low all the time, because the Mississippi River carries so much excess fertilizer from that farmland that it dumps into the ocean there and stays and causes some major problems. And I'm sure you've seen or heard about red tide bloom off the coast of Florida is very common. So yeah, that's pretty much all you need to know about the nitrogen cycle and nitrification.